Okay, just want to welcome everybody for our OCB webinar today. I'm going to give a few introductory remarks and then I will pass the baton over to May, who will be sharing the presentations. So welcome. My name is Heather Benway. I manage the Ocean Carbon and Biogeochemistry Project Office, which is hosted by Woods Hole Oceanographic Institution. The OCB Project Office includes two other staff members, May Mahegan, who manages OCB Communications, and Mary Zawoski, who manages Administrative and Logistical Operations. I'd also like to acknowledge the sustained support of NSF Chemical and Biological Oceanography, as well as NASA's Ocean Biology and Biogeochemistry programs, which make the OCB program possible. Today, we're joined by Amy Moss. She is from the Bermuda Institute of Ocean Sciences and a member of the OCB Scientific Steering Committee and Export Science Team, and she'll be serving as my webinar co-host today. Just to give you a sense of the scientific breadth of OCB, these green icons depict our overarching science areas. And you can find more specific research questions of community interest on our website under the About Us page. We definitely invite you to engage with the OCB network in all of the ways listed here. Please visit our website, sign up for our biweekly e-newsletter, and/or follow us on social media. And you can always reach us by email at the address listed here. So today's webinar is all about exports. The format of the webinar is a little different from previous webinars. First, note that this is a 2-hour webinar. This is really an extended time period to share the wide range of science that exports has to offer, and also enable plenty of time for Q&A with our audience. The webinar is being recorded for posting on our YouTube channel, and we've broken up today's webinar into 4 parts, separated by 3 Q&A sessions. The webinar will start with a brief overview of the Exports Field Campaign by Dave Siegel. He is the Exports Science Lead, and then we'll follow with a series of vignettes describing the research being conducted by members of the Exports Science Team, including a number of early career scientists today. Woohoo! Each of the speakers will introduce themselves at the beginning of their talks. The first set of vignettes will focus on plankton and productivity measurements in food web synthesis, and the second set of vignettes will focus on particle fluxes and mass balance calculations. Dave will then wrap us up at the end with a brief update on the field campaign and closing remarks. Given the number of talks and presenters today, all of today's presentations were pre-recorded, and May has heroically assembled all of them into the movie files that we'll be viewing today. All of our presenters are here with us today as panelists. Amy and I will be moderating the 3 10 to 15 minute Q&A sessions today. When you type in a question, please note if it's for a specific speaker or if it's a general question for the panel. If we don't get to all of your questions for the speakers, we will definitely share them with the speakers so they can follow up with attendees after the webinar. Here's a quick screenshot of the attendee WebEx panel, which changes on a daily basis, so I'm sorry if this isn't exactly what you're seeing. Please note that all attendees are muted with video off just to reduce background noise and distraction. On the bottom right is the chat function, is that little messaging speech bubble icon, and if you look at the ... right next to it, to the right of it, you can find the Q&A function in there. You can use either of these mechanisms to pose your questions. When entering your questions, I have enabled the chat this time so that your questions will automatically go to everyone, just to ensure that nothing is lost. Looking forward to a great series of talks today.
Hello, everyone. Today I'd like to talk about an update about the progress and challenges for the Exports Field Program. Export stands for Export Processes in the Ocean from Remote Sensing. It is a NASA field campaign. The presentations will be made by the Export Science Team. My name is David Siegel. I am the Export Science Lead and a professor at UC Santa Barbara. So what will follow is I'll give a short presentation on the goals and objectives of the Exports Program. I'll talk about what we did operationally in the Northeast Pacific, and then this will be followed by 9 science vignettes given by members of the Export Science Team. So What is the goal of the export science team? The goal is to predict the export fate and carbon cycle impacts of ocean net fire production from satellite observations on regional to global scales. The focus here will be on quantifying export pathways and NPP fates over a range of ecosystem states. Our emphasis is on to develop our predictive understanding of these processes is by measuring and modeling the regulated processes. Exports has leveraged recent advances in optical oceanography, single particle imaging, genomics, autonomous tools, and many other things. So let's talk about pathways. The important thing, the important conceptual thing to understand here is we have two ecosystems. We have an ecosystem in the euphotic zone where phytoplankton fix CO2, make net pipe production and fuel the euphotic zone ecosystem. There is export from that process from the surface ocean to depth through a variety of pathways. That supplies the energy for the twilight zone ecosystem as shown here. So our focus is on pathways. And there are three major ones to focus on. There's the sinking particle flux, both by sinking aggregates, fecal matter, or intact phytoplankton. There is the migrant pump, where Mesozooplankton migrate up and down vertically on daily timescales, up to the surface at night to feed, and down to depth to avoid predation. That leads to an active transport of carbon from the surface ocean to depth as the zooplankton respire and excrete carbon at depth. And the last transport mechanism is the physical injection pump, and this is where suspended carbon and DOC are pushed down to depth where it is utilized by the community below. This might happen on global scales due to the overturning circulation of the global ocean, or it could happen on some mesoscale frontal scales. Once that carbon's down there, it is utilized by the twilight zone ecosystem, and the rate at which the twilight zone ecosystem feeds on this organic matter that controls the scale, the vertical scale by which the upper ocean affects the carbon budget at depth. So exports has three major science questions based upon this. The first is, how do upper ocean ecosystem characteristics determine the vertical transfer of organic matter from the euphotic zone into the twilight zone? What controls the efficiency of vertical transfer within the twilight zone, and how can we use this knowledge to reduce uncertainties in present and future estimates of exported feeds in NPP? So to do this, we are sampling over a range of scales. On the left is conceptual food web diagram for the Northeast Pacific at Station P. We sampled this in the fall of 2018, and that is the focus of this of the talks to follow. Station P is a rather inefficient exporter per unit net price production. Its export flux is low compared to what's in many regions globally, as shown on the x-axis in this diagram from Boyd and from Bissler and Boyd. On the x-axis, it also shows that the material, once it gets fluxed out of the surface ocean, is rapidly utilized at a low transmission rate by the ecosystem in the twilight zone. Whereas in the North Pacific, which we had planned to do this year and will hopefully do this in the spring of 2021, is a much more efficient exporter of net production energy, and the flux transmission is much lower. Excuse me, the flux transmission is much higher through the twilight zone. So exports to project to people. There are 19 projects, 56 PIs, at least that many students, postdocs, technical staff, etc., who are participating in the project. Why do we need so many people? Well, we need to understand all the ways in which these pathways function and how we sample them. So that means from a systematic area of science, we have to understand genomics, microbiology, physiology, zooplankton ecology, physical oceanography. From a sampling point of view, we have to understand robotics and bioinformatics and biooptics and single particle imaging and characterization. So the exports project in North Pacific was conducted like this. We had a process ship, with a Roger Revell, that measured rates and stocks following a Lagrangian float, which is shown here. The Lagrangian float was drove at depth, and it followed the water masses there. by following the Lagrangian flow, we did the rate sampling done on the Van der Velde was done in a quasi-Lagrangian fashion. The Sally Ride was the survey ship. It made surveys around the process ship in the Lagrangian float, and then we augmented everything with a set of autonomous vehicles that operate on a range of time and space scales. We had a sea glider. There's the instrumented Lagrangian float. There's a wire walker. The BioLargo. We had several BioLargo arrays. And we deployed nutrient sediment traps and a sediment trap array to directly sample vertical fluxes. We partnered with the Canadian Line P program, as well as used data from the NOAA's PMEL Ares interaction mooring, and used data from the OOI global node. So what do we know about this site? Well, Station P is a high nutrient, low chlorophyll site. Duh, we could have figured that out by just looking at the literature. And at high nutrient levels, low chlorophyll, and low dissolved iron concentrations as expected. Actually, the chlorophyll levels were a bit lower. The photic zones were a bit deeper than had been seen from the Line P climatology. Phytoplankton and net migration were dominated by smaller autotrophs, but we had strong top-down controls, zooplankton mediated controls on food web function and vertical fluxes. This led to a low export efficiency and rapid attenuation of sinking carbon fluxes with depth. And the last major characteristic to talk about is that it was an eddy desert. The subinertial current, there was very little mesoscale energy. Subinertial currents were about 2 centimeters a second, about 2 kilometers a day. But microscale water masses were apparent in our data sets. So the timeline kind of looked like this: the two ships, the Roger Elval and the Sally Ride, left in mid August and came back in mid September of 2018. They sampled these 3 major 8-day epochs, which are shown by the vertical bars here. Embedded within the process study was a set of robots. We had a sea glider that we deployed during the OOI turnaround cruise. And during the cruise, we deployed our Lagrangian float, a series of BioLargo floats, one of which is still operational. And then 2 weeks after we had left it, we went home, the Line P program went out and sampled our same sites. All of the autonomous vehicles were picked up in a recovery cruise in December. So the process ship, so these are the 3 elements I'm going to go through. The process ship assembled in these 3 8-day epochs. Its focus was on rate, flux, food web, composition determination, following the Lagrangian float, enabling the measurements to be made in a quasi-Lagrangian sense. Sampling included trace metal clean and standard rosette cast, continuous underway analyses through a variety of tools, mockness and zooplankton net tows, ring stone catcher deployments, and optical profiles. The survey ship conducted spatial surveys around the epoch. The years are shown in the top row in large scale, and in the bottom row in small scale. What was sampled and how was sampled following during each of the 3 epochs. It provided spatial variations in optical physical export, net community production, plankton composition, particle size distribution things. It supported the process ship because that's where the high volume particle pumping was done, and it acted as an intercalibration source between the autonomous array and the process ship observations. So the autonomous array was a series of instrumentation listed in the second row, but it extended the spatial and temporal scales of the observations. All data were intercalibrated with the ship data and among the suite of AUVs. It extends the observations from July 2018 to the present with the BioLargo focus going there. The top row here is shown BioLargo results for nitrate, chlorophyll, and backscatter, and particle backscatter, and then shown on the far right column, you can see that you can look at the variability of backscatter on a variety of temporal scales. Here is shown, on this sort of annual scale from BioLargo floats, on a more monthly scale from the sea glider, and then on a daily scale from the wire walker. So what will follow is a series of 5-minute science vignettes given by members of the export science team. I'm not going to go through and read this list here. Maybe it's time to take questions. Thank you very much.
Okay, let's start with a round of questions anybody might have on the exports field campaign for Dave or other members of the panel. Remember, you can use the chat or the Q&A. It may be that Dave has explained it so many times to this community that they're all experts now. Experts on exports.
Yeah.
Not a problem. That leaves more time for questions about the vignette.
Exactly. All right, let me just make sure there's nothing in the Q&A here. Oh, we have it. Cheeky question. Can we get t-shirts with that great project schematic?
I'll stick it in the chat. I've got to find it. It's my daughter actually made this for her printmaking class. So we have t-shirts and hats and whatever, all sorts of silly things. I'll send it along. I just got to find it.
And I have to share that they have cool zipper pouches too. I have one.
We got everything actually.
Yeah. I guess we do have some swag. I'm trying to get her to do one for the North Atlantic. We'll see how that goes. And I apologize for listening to me stutter. It's awful. But
Dave, we love listening to you give talks.
Oh God.
Especially when you're talking about something as cool as exports. All right, last call for questions, and then we'll move on to the vignettes. All righty. I'm going on to mute, and I'll let May have the floor again.
Hi, my name is Jason Graff. I'm an assistant professor at Oregon State University. And today I'll be presenting to you some work that my colleagues and I have been doing to characterize the phytoplankton community during the NASA Exports Field Campaign in 2018 in the North Pacific Ocean. To do this, we're really interested in a number of community composition metrics, including abundance and diversity, size, shape, and taxa-specific information. All of these metrics help set the stage for the biological bump and our understanding of it. To characterize the community, we're using multiple approaches, including HPLC pigment analysis, multiple cytometric approaches, and high-throughput sequence analysis. Each approach provides unique data set, and combined can provide even richer, more complementary information than any single approach alone. Here's an example of how two complementary approaches can be combined to give us a better sense of the phytoplankton community. Here we've merged the size calibrated data from the standard flow cytometer with that of the imaging flow cytometer to provide a full phytoplankton size spectrum ranging from just smaller than 1 micron to almost 100 microns. And this data is really critical because from it we can actually start to calculate biovolumes and carbon estimates for very specific size categories. We can also see that small cells were very abundant during this study. From the flow cytometer, we know that Synechococcus and picoeukaryotes were the most abundant cells. But we also can see from the imaging flow cytometry that large cells were also present and possibly played critical roles in this environment. The figure on the left shows a typical depth profile of the groups that we can identify in the standard flow cytometry. These include Synechococcus, nanoeukaryotes, and picoeukaryotes. We can see that Synechococcus dominated throughout most of the water column by abundance, followed by picoeukaryotes and then nanoeukaryotes. The figure in the center shows the biovolume contributions for each individual size bin shown on the previous graph. From this, we get a sense for the relative contribution of these different size classes and can look in very fine resolution at what each size class contributes. For even finer resolution, we can look at the size classes to the right of that dashed line, or approximately 7.5 microns. These cells were imaged by the imaging flow cytometer and give us a sense for which groups actually and which taxa provide the biomass within these size classes. The figure on the right now displays the results for the imaging flow cytometry at different depths. First, we can see that the total biovolume is decreasing with depth. We can also see that the dinoflagellates, shown in green, represent a significant proportion of the biomass at each depth, while diatoms and other taxa are present, but contributed significantly less to the total biovolume.
Hi, my name is Sasha Kramer, and I'm a PhD candidate with Dave Siegel at UC Santa Barbara. Here, we're looking at the results of phytoplankton diversity in the euphotic zone, measured via high-throughput sequencing of the V4 region of the 18S gene. The major groups that emerge from this analysis are dinoflagellates, which have by far the highest relative sequence abundances, diatoms, and prymnesiophytes. Similarly to the IFCM data, dinoflagellates are very well represented in these euphotic zone samples. However, when we look at the accessory pigment data, here is shown ratios of pigments to total chlorophyll A, a slightly different story emerges, where low concentrations of paradennin, a dinoflagellate marker pigment relative to chlorophyll A, would indicate a lower presence of dinoflagellates compared to the other two methods that have been discussed so far. These are also all surface samples, and fortunately we see that the diatom and prymnesiophyte accessory pigments are well represented in the HPLC pigment data set, similarly to the representation of those taxa in the IFCM and high throughput sequencing data sets. The automated IFCM data enables us to look at the spatial variability of different taxa, but also consider variations in cell size. So here, looking at dinoflagellate biovolume in the surface measured on the survey ship, as well as a composite mean of all dinoflagellate cell sizes measured over the course of the cruise. We can also look at diatom biovolume, which similarly shows high spatial patchiness, but very different patterns and abundances compared to the dinoflagellates. Diatom cell size distribution is also quite different than the dinoflagellates. The high resolution of this imagery data allows us to dig more deeply into some of the variability in cell size. Using the dinoflagellates as an example, the IFCM data enables us to look at different cells, such as these very small ones that are only a few microns across. Examples such as these oxytoxum, where at least two species comprise really different size distributions in the overall data. And then these serratium cells, which have very diverse size distribution, but for which the equivalent spherical diameter is not really an appropriate marker of cell size, given the different orientations that can be measured of these cells. There are also numerous other dinoflagellate taxa that were measured, but these are just some examples of cells that have very different sizes and distributions over the course of the cruise.
Phytoplankton community structure during the Export's North Pacific Field Campaign was fully realized through the use of multiple complementary approaches, including HPLC pigment analysis, flow cytometry approaches, and high-throughput sequencing. We found that phytoplankton were abundant and formed a relatively consistent background community in the surface, but also showing increased abundances below the mixed layer. Spatial patchiness was observed in the diatoms and dinoflagellates using the imaging flow cytometry. And these cells were also found to have a larger footprint throughout the phytoplankton size spectrum than previously realized. More importantly, the size metrics of these cells were not fully resolved or described using the equivalent spherical diameter as the metric of interest. Thus, we need to consider other metrics which more fully represent the community and their potential role in the export of carbon to the deep ocean. A high abundance of dinoflagellates raise the question, what is their role for carbon export? If these cells are autotrophic, mixotrophic, or heterotrophic, what can we learn about their role in the biological carbon pump? Thank you, and we'll leave you with some additional acknowledgements for people who collected samples and provided data for this presentation.
Hello, everyone. I am Meredith Meyer, a graduate student in Dr. Adrian Marchetti's lab at the University of North Carolina at Chapel Hill. Melanie, Feen, Shannon, Burns, and I will be talking about productivity trends at Station P. From the North Pacific cruise, we have numerous in situ productivity measurements, as shown in the figure here, as well as dissolved iron concentrations, which are crucial to explain productivity trends in this high nutrient, low chlorophyll region. Funding for this work was provided by NASA and the NSF Division of Ocean Sciences.
Hi everyone, my name is Shannon Burns and I'm a graduate student in Dr. Kristen Buck's lab at the University of South Florida College of Marine Science. During the cruise, a towfish was used to collect seawater from about 2 meters deep for dissolved trace metals, which are micronutrients that influence primary production at Station P. Dissolved iron concentrations were very low in surface waters, averaging about 30 picomolar, and were generally less than 100 picomolar through the entire euphotic zone of the water column. A series of grow-out experiments limitation of phytoplankton in these waters, with iron additions resulting in large increases in biogenic silica and chlorophyll A relative to controls. Observations via IFCB and amplicon sequencing indicated that it was diatoms, specifically pseudonichia, that dominated the growth response when iron was added with macronutrients.
In measuring primary productivity from carbon-13 incubation experiments, we aim to assess general net primary productivity trends, size fractionated trends, and the balance between new versus regenerated production. All of these metrics relate directly to transfer efficiency and carbon export. Firstly, we saw a clear indication of a small cell community through the predominance of the less than 5 micron cells in the net primary production, where it accounted for 60% of the total rate. Based off of new and regenerated production, we calculated an F ratio consistent with previous estimates at Station P, and indicating that Station P is a recycled nutrient community dependent on regenerated production. Because of the dominance of small cells, we observed a close coupling between the total NPP and the less than 5 micron NPP over the course of the cruise, with a peak mid-cruise. Additionally, we observed a corresponding peak in our new production, regenerated production, biomass data, and assimilation rate data, indicating what appears to be a period of increased biologic activity.
Hi everyone, I'm Melanie Feen. I'm a graduate student at the University of Rhode Island working in Dr. Melissa Oman's lab. And so my research has been focused on interpreting the wire walker observations collected during exports. The wire walker, it's an autonomous profiling drifting sensor platform that traverses the upper 500 meters and back every 40 minutes. It's quite a rapid profiling platform, and because of its high frequency and water following nature, we can begin to tease apart biological and physical interactions from hours to days. Here I'm highlighting the temperature and salinity variations within the mixed layer, and here's the beam attenuation particulate organic carbon proxy. So zooming in to August 27, which coincides with the day of high productivity as shown by Meredith on the previous slide, we take all of the particulate organic carbon proxy in the mixed layer and fit a Diel cycle following Brown et al. 2019 to determine the gross primary productivity and the community respiration loss term. So zooming out to the entire time series, the wire walker is in green and the glider oxygen-based productivity is provided by Rue Nicholson and Shiny Trailer, and that's in purple. So we can see the productivity peak on the 27th, which was coupled by also a peak in respiration as well. Considering the wire walker beam attenuation, particulate organic carbon proxy, and the glider oxygen-based Diel productivity are independent platforms using different metrics to compute these estimates, they're in generally good agreement with each other, and also with the temporal trends Meredith showed on the previous slide from incubations. So with this in mind, we would just like to end on a summary that the grow-out experiments confirmed iron limitation during exports, consistent with our expectations for Ocean Station Papa. Incubation results are further consistent with iron limitation in the system, as we observe low primary production driven by small cell regenerated production. And when looking at Diel productivity from in situ sensors, these independent estimates found similar temporal trends to the incubations, and the wire walker Diel productivity showed a similar magnitude to the glider-based method. Thanks for listening.
Good afternoon, everyone. My name is Karen Stamieszkin, and I am a postdoc at the Virginia Institute of Marine Science. I'm going to talk to you today about zooplankton-mediated pathways of carbon export, circled in red on the diagram on the left. The left-hand arrow in that red circle indicates the passive flux pathway, which consists of sinking fecal material from zooplankton feeding at the sea surface. Passive flux can also include sinking zooplankton carcasses. The right-hand arrow in the red circle represents the active flux pathway, which consists of carbon that is actively transported by zooplankton who feed at the sea surface at night and then migrate to depth and metabolize that prey during the day. At depth, these zooplankton breathe, poop, pee, and die, all contributing to active carbon flux. Moving on to methods, we conducted day-night pairs of mock nest net tows to 1,000 meters to quantify the zooplankton stocks at Station P. We also conducted shipboard experiments to measure fecal pellet production rates, respiration rates, and excretion rates of dissolved organic carbon in collaboration with Craig Carlson. Finally, we combined our stocks and rates to estimate the strength of the active and passive carbon flux pathways. The vertical distribution of zooplankton at Station P showed a typical pattern of increased biomass in the euphotic zone at night compared to the day due to Diel vertical migration. Shown in the larger plot is biomass by size fraction with daytime values on the left and nighttime on the right. Diel vertical migration resulted in a 2.5-fold increase in zooplankton biomass in the euphotic zone at night. You can see in the acoustic data that zooplankton distributed in distinct bands at night and during the day as they migrated up and down in the water column. We classified zooplankton not only by size, but also taxonomy. We found a dense layer of Neocalanus cristatus, a large calanoid copepod, migrating from about 75 meters to the surface, an unusually short Diel vertical migration. These guys made up the majority of the 2 to 5 millimeter size fraction in the upper 100 meters. This size fraction is shown in lighter blue on the plot. We also found Salpa aspera, a gelatinous pelagic tunicate, migrating from the 4 to 500 meter depth stratum to the surface daily. It's represented in yellow. For context, biomass at Station P was about 65% of that measured in the Northwest Pacific a little earlier in the season during the Vertigo expedition, which took place several years ago. This difference is probably in part due to iron limitation of primary production at Station P. We measured rates needed to estimate passive and active carbon flux through the zooplankton. These included particulate organic carbon production in the form of fecal pellets, inorganic carbon production as respiration, and dissolved organic carbon production as excretion. Similar to biomass, we took both taxonomic and size-based approaches. On the left, you can see that Neocalanus cristatus had relatively low individual fecal pellet carbon production rates, while salps had exceptionally high rates. And for your viewing pleasure, you can see those organisms alongside their very different species. You can see that salp fecal pellets are much larger and therefore have a higher carbon content than Neocalanus pellets. The middle plot illustrates relative contributions to biomass and fecal pellet carbon production in the euphotic zone by the different zooplankton size fractions during day and night. Note the disproportionate contribution by the smallest zooplankton shown in white to fecal pellet carbon, as well as the contribution of salp's fecal pellets at night. The plots on the right show allometric relationships for zooplankton respiration and excretion calculated using experiments with different taxa. Okay, now we bring it all together by applying our experimentally derived rates to the measured zooplankton biomass. The left-hand plots show active flux to the twilight zone or below 100 meters. Again, this carbon is transported by migrating zooplankton who feed at the surface at night and then metabolize at depth during the day. Here we have separated crustacean mesozooplankton and salps because of their very different allocations of carbon. The majority of active carbon export facilitated by crustacean mesozooplankton was from respiration and estimated mortality at depth. Meanwhile, salp active transport was mainly attributed to fecal pellet production at depth. For passive transport, we were able to compare our measured fecal pellet carbon production rates with flux rates from fecal pellets caught in sediment traps. These data from Colleen Durkin's sediment trap analyses show that the majority of passive fecal pellet flux out of the euphotic zone was due to sinking salt pellets and pellets from larger macrozooplankton, which we did not include in our experiments. So while we measured a large relative contribution of fecal pellet carbon production by small zooplankton, these pellets were mostly attenuated in the euphotic zone. On average, we found that active carbon export was about 40% of the passive export, but looking at the range of the two pathways, it is clear that they are comparable in magnitude. I want to end by emphasizing the outsized role of SALPs at Station P during the exports experiment. We found passive carbon flux from SALPs was similar in magnitude to active flux, and that even SALP carcasses estimated to be sinking out of the euphotic zone were a significant source of carbon export. The maximum active and passive carbon export from SALPs was on par with average total carbon flux measured in sediment traps. On the right, you can see from Colleen Durkin and Megastappa's sediment trap data that while ephemeral, SALPs can produce nearly all of the sinking carbon down to 500 meters. Their ecology is such that they can take advantage of the small-celled primary producer community typical of high-nutrient, low chlorophyll regions, and can also persist in boom-to-bust food webs typical of highly seasonal systems. So, we suggest that the impact of this pelagic tunicate on the carbon cycle of this region is perhaps underappreciated and very important.
Hi, my name is Brandon Stevens, and I'm a postdoc in Greg Carlson's lab at UC Santa Barbara. I'll be joined later by Garrett Sharpe, who's a PhD student in Scott Giffords' lab at UNC Chapel Hill. Together, we'll be presenting observational and experimental data on the dynamics of bacteria and dissolved organic matter from the export Station P cruise. As shown in the image on the left, there's a direct connection between the DOM reservoir and the microbial food web, where DOM consists of a small yet highly reactive pool within a much larger, less reactive reservoir. We begin by presenting mean mixed layer dynamics of net bacterial production and bacterial biomass, both of which exhibited systematic temporal patterns. For instance, there was a nearly twofold increase in bacterial activity that began toward the latter half of the cruise. Concurrent with this bacterial activity, there was a nearly twofold increase in the concentrations of chlorophyll A and a steady decline in nitrate-nitrite. These trends in chlorophyll resembled the concentrations of POC. However, both DOC concentrations exhibited little resolvable systematic variability. Instead, the relative contribution of total hydrolyzable amino acids, which can be used as proxies for the reactivity of DOM, suggested that a more labile DOM began to accumulate as bacterial activity increased. Overall, mixed layer patterns demonstrated that an increase in phytoplankton biomass and labile organic matter coincided with increased bacterial activity, suggesting that there was a link between DOM quality and microbial response.
Hey everybody, this is Garrett Sharpe from the Gifford Lab, and I'm going to briefly talk about the bacterial metagenomes we got from the mixed layer at 5 to 12 meter sampling depth over the course of the cruise. As Brandon has previously stated, bacterial productivity shows clear shifts across the course of the cruise, and we can see that there is a similar pattern present in the bacterial metagenomes based on the PCA plot below, where there's a distinct community state in epoch 1 that then shifts into a new community state into epoch 2, and then shifts again into a new community state in epoch 3 that looks a little bit more similar to the state in epoch 1. We also see this pattern in the absolute bacterial abundances for various bacterial taxa, where, for instance, with Pelagibacteriaceae, there's a high cell abundance followed by a crash in the cell abundance in the beginning of epoch 2, and then a slow to fast recovery depending on the taxa through epoch 2 into epoch 3. And we can see that this pattern persists for many of the bacterial taxa in the high abundance taxa group, moderate abundance taxa group, and a few of the members in the low abundance taxa group.
To quantify DOC removal and bacterial response over timescales of days to months, we conducted DOM remineralization experiments during the cruise. These dilution cultures consisted of mixing 30% of a 3 micron pre-filtered inoculum with 70% of a 0.2 micron filtrate. This design effectively reduces grazing pressure on bacteria and allows us to isolate the interactions between bacteria and DOM. Results from 3 mixed layer experiments shown here demonstrate the DOC removal increased later in the cruise and was greatest in that last experiment shown in green. The degradation index of the initial DOM was also elevated in the last experiment, indicating that a more labile DOM could have influenced the elevated overall DOC removal. Regressing the initial DOM composition with DOC removal rates for all experiments conducted during the cruise resulted in a significant relationship, suggesting that the degradation index could serve as a useful indicator for the removal of DOC by bacteria at station P.
We also took metatranscriptome samples from the remineralization experiments Brandon just spoke about, and we found that for most bacterial taxa, the relative abundance of transcripts does not change much, with a notable exception of this group in pink, the Pisca-Rickettsiaceae, which are known methylivores. So it appears that methotrophy could be increasing over the course of each experiment to varying degrees, depending on the experiment. It should also be noted that although relative abundance of transcripts does not change for most of these taxa, that doesn't mean that the functions transcribed by this transcript pool doesn't change over the course of each experiment. And so in the future, we're going to be looking into what functions are expressed in each of these time points to see if changes in expression of different functions can explain why there's low DOC drawdown in these first 2 experiments and high DOC drawdown in this 3rd experiment, E2R3.
Hello, my name is Sarah Lerch, and I am currently a postdoc in Dr. Bethany Jenkins' lab at the University of Rhode Island. Today, I'm really proud to be representing the collaborative efforts of the Exports Food Web Writing Group. The goal of this group is to synthesize the really diverse data sets that have been collected by the working group members listed here in order to better understand the food web and its relationship to carbon export. So this is an effort that's being led by Heather McNair, with contributions from myself, as well as other co-writers James Fox, Meredith Meyer, Amy Moss, and Tatiana Rainierson. So our group is really focused on 3 main goals. First, we want to use the food web to describe carbon cycling at Ocean Station Papa. And this is really focused on carbon associated with stocks and rates. Ultimately, we hope to do this for 3 separate depth layers, but today I'm just going to be talking about the mixed layer. Second, we aim to assess carbon budgets for key stocks, including zooplankton, phytoplankton, and bacteria. And finally, we hope to generate broader hypotheses about how environmental factors influence carbon flow within and export from these food webs. We plan to do this by looking at how stocks, rates, and flux vary from the cruise average. So here I'm showing a really simple diagram of the 3 carbon stocks associated with a marine food web. We have carbon dioxide, particulate organic carbon, and dissolved organic carbon. And I'll use this diagram to fill in the stocks and rates, which were either directly measured during the cruise or can be inferred from measurements that were made. So in addition to measurements of POC and DOC, stocks within and overlapping POC were measured, including different size fractions of phytoplankton, zooplankton, and bacteria. We also have rates which influence these stocks, such as those associated with primary production, zooplankton grazing and digestion, bacterial carbon demand, and of course, particulate organic carbon flux. These rate arrows are all associated with average rates during the cruise and have been scaled to the largest rate, gross primary production, in order to help visualize a given stock's balance. As you may be able to see, many of the in and out arrows for a single stock are similar in size, indicating a relatively balanced system. But another way to look at this balance is to sum up the rates of carbon coming into and moving out of a given stock. So we've done this here for microzooplankton, phytoplankton, bacteria, and mesozooplankton. And as you can see, these inputs and outputs are similar magnitudes, again, indicating a balanced system. But The variability associated with these measurements also indicates that there are fluctuations in these rates during the course of the cruise. So we're really interested in looking at these variations in order to better understand the connections between environmental parameters, stocks, and rates as the food web fluctuates around that cruise average. So in order to identify potential patterns, we visualize the anomalies of rescale data from the cruise average in the form of heat maps. And here you can see that temperature, particulate organic carbon, and net primary production all increased relative to the cruise average in the second half of the cruise, for example. So this really provides just a first peek at the types of data and trends we're going to be able to visualize with this approach. Ultimately, though, the big goal and what we really want to do with this is to better understand the relationship between food webs and carbon export at Ocean Station Papa. So one of the big questions we're tackling to get at this is what influenced carbon transformation in the system. Here we are applying that anomaly approach I've shown you across all of our data sets to get at this. Another big question is who's transforming carbon in the system? There's multiple groups that are using sequencing as well as imaging approaches to answer this question. This is going to give us an idea of what community members were actually present at the site. The answers to these questions, when combined with those measurements of carbon flux, will enable us to better understand the relationship between the food web and carbon export at OSP. So with that, I'll thank you for your attention, and I welcome any questions once the full presentation has been finished.
All right, so now we're at the point where we're welcoming questions on those first 5 vignettes. So if you have questions, again, please either put them in the chat or in the Q&A, and everybody's online and ready to respond.
Questions for our speakers? Oh, I see something. Here's a question. Could the dominance of dinoflagellates in the sequence data be a result of multiple copy number in this group?
Yeah, I'll tackle that. Absolutely. I think that's always an issue when we're talking about dinoflagellates and the number of copies that they can have. And I think it's something we're just starting to look into with this data set. And I think once we've got a handle on that, comparing it again to the different analyses that we have from the imaging flow cytometry and the pigment community composition analyses that we're going to do will be a nice comparison.
This is Heidi Sosik. I'll just add that there's no doubt from the images that there were a lot of dinoflagellates. So the mystery I think is actually why the marker pigment for dinos appears so low relative to what we see in the imaging, and given the caveats, what we see in the sequence results.
I can answer this question in the Q&A about HPLC pigment analysis. We have not tried chemtax, but we're using some other pigment-based methods that are less prescriptive in how they allocate different accessory pigments to different groups. So there will definitely be future exports-related presentations that explore pigment data in that way, but we do not plan to use ChemTax.
I've got a question here for Jason Graff. Remind us of what was the depth variation of the Phytoplankton abundance. You mentioned an increase with depth. That is the absolute or relative sense, and why is that happening?
I can answer that. In talking about the pico phytoplankton, what I saw was actually an increase in Synechococcus with depth, and they really increased significantly from the surface mixed layer down to about 60 meters, at which point they started to drop off again. And so that was in very absolute terms that they were increasing at that depth.
So for those of you who are asking questions, I think there are questions coming in the chat that are going to all attendees. Please note that the panelists and the host cannot see those. You need to select to everyone in order for us to be able to read those questions to our panelists. So if you've asked a question in that manner, please ask it again so that we can make sure that they're answered.
This is Heidi again. I see that there's a question about whether the dinoflagellates were heterotrophic. I think that's a little bit tricky to be definitive about, but the fluorescence that we see per cell from the imaging flow cytometry suggests that most of them were either mixotrophic with some autotrophy ongoing or autotrophic. They didn't appear that there were a lot of dinos with low fluorescence relative to their size. Certainly there are some morphologies in taxa that were heterotrophic, but I don't think right now that those were dominating the biomass. Of course, dinos are, notorious for being mixotrophic too.
Okay, I have a question for Jason on investigating mixotrophy. Have you attempted to investigate food vacuole ID and low-high fluorescence with the dinos? Could the mixotrophy be a reason why the pigment's low?
I think Heidi's probably more equipped to discuss the fluorescence of the dino, which she just addressed a little bit, I think.
Yeah, that's what I was trying to say, that like if we consider biovolume, fluorescence per biovolume, it doesn't look like they're heavily heterotrophic, but it's, they can have fluorescence because their guts are full of fluorescent food. So it's tricky.
I'm wondering about the relevance of your findings for harnessing the ocean for active carbon dioxide removal, which is of increasing interest for CO2 mitigation globally. Anyone want to tackle that?
Yeah, I think I should. There's, in some sense, we're studying how the natural system is working. There is no management action that we are talking about here that would allow any sort of CDR strategy to be employed. I mean, understanding the base state is important. There are CDR strategies in terms of iron addition and those sorts of issues. The idea in exports overall is to build up over a range of states. How does the biological pump change? How does it change as a function of of global warming, but other things as well? So there, as we'll talk about, I mean, the idea is to put together these data sets so as we can develop models that will test these sorts of things. I hope that was clear.
Interesting to see spatial variability in large phytoplankton in absence of strong eddy front activity. Were there any horizontal spatial correlations or anti-correlations between groups?
I can answer this a little bit too. We are looking at this both in the pigment data sets and shortly in the IFCB data set. We do see some changes in the pigment suite moving around by looking at sort of hierarchical clustering and that sort of analysis. I think it is super interesting. From a system that is top down, I would not expect everything to be driven by physical processes, which are largely bottom up. So we will see how this works. We are just at this. We are just starting this kind of analysis, Adrian.
A question from Pedro Tura. A more general question, do you think the deep chlorophyll max should get a special approach? If so, how are you parameterizing them in the exports project, as they are not measured by remote sensing?
I would say that part of our analyses, which we show everything today, is that we are that and trying to draw links between the surface mixed layer and what we see below the mixed layer. The direct carbon measurements of phytoplankton biomass show that there is an actual increase in biomass at depth, but the increase in chlorophyll is also partially due to photoacclimation. And so teasing apart what is biomass and what is physiology is part of the analyses that we will be looking into as we move forward with the data set.
There's a question asking what is the pre-filter size for IFCB? Can Jason or Heidi address that?
Yeah, I was just trying to type, but I can speak type, I guess. Typically, we use a 150 micron pre-filter at the entrance to the IFCB to prevent clogs. Cells that are longer than that do get through and get imaged and quantified. As long as they're long and skinny, they can go through the filter and be imaged several hundred microns long. But there is, of course, questions about the efficiency of which we sample those because of the pre-filter.
Thanks, Heidi. A question from Alexander Bogdansky. How will you model the episodic nature of large fluxes caused by SELP blooms?
God, I don't want to touch that.
The $100 million question.
I think that's a really interesting challenge because a lot of models have been based on what we know about crustacean zooplankton, and SALPs, while their biovolume doesn't really fit on any of the sort of metabolic allometric relationships that people generally use for modeling, their carbon content does, because they all of a sudden are smaller. So they can feed on really small things and therefore are episodically successful. So I think that considering them by their carbon content rather than their actual biovolume will enable them to be added to models that we already have. That would be part of an answer. Does anyone want to add to that?
Thanks, Karen. Does anyone have anything to add? All right, another question. For the DOM lability experiment, what is the reason for the August 25th sample on which you saw higher lability from exudate of phytoplankton or bacteria?
Yes, hi, this is Brandon. I tried to answer this briefly in the chat, but basically we see a nice increase in lability indicators based on our amino acid analyses. So we ended up performing regressions with all of our experiments and found that this held across both time in the surface and depth, where increased lability based on amino acid analysis was associated with elevated DOC removal.
Other questions? Am I missing any questions? Last call.
Yeah, I just have one thing to add to the answer that Karen did on how we would model SALPs. I mean, when we've considered exports, when we were working on the planning, we realized that it was not necessarily everything would come from remote sensing. And so to at least to characterize the, I mean, there is hopes of these imaging devices like the UVP, which we'll talk about shortly, that if we had an array of these things globally on bioargo floats or biogeochemical imaging floats, I suppose you would call them, well, there could be data sets to do this. Because the hard part, because SALPs are so ephemerally, spatially, and temporally, how we do that modeling does become pretty difficult. But I think there's tools on the horizons that will allow us to do this, and that's, if that's one of the answers, that comes out of all this, that's a good thing.
To add on to that,
Okay.
not only the imaging, but also some of the molecular eDNA type things might help us track the importance of SALPs in a similar kind of way.
I've got one more question here. What caused the variation of Pelagibacter abundance among 3 epochs?
So I think that the reason that the Pelagibacter may have dropped was just simply due to the lower bacterial productivity during that time period that probably goes along with the lack of resources to provide for high bacterial productivity. Although it's not entirely clear, it's also possible we just moved into a region that just had lower abundance. Though if we stayed in one water mass, it's probably just due to lower resources.
Okay, why don't we move on to the next set of vignettes? If people come up with questions while we're transitioning, we can address them after the next set.
Hello, my name is Andrew McDonnell from the University of Alaska at Fairbanks, and on behalf of all my collaborators on the screen here, I'm going to be talking about particle size distributions from optics. But before we get started, fun fact in preparation for my 5 minutes of OCB webinar fame, I actually cut my own hair this morning, so maybe let me know in the comments if I missed a spot or something. And I noticed that some of the exports team is actually looking a little scruffy around the edges, so I'm more than willing to schedule a one-on-one Zoom consultation with you to walk you through my strategies for cutting my hair. Anyways, back to the topic at hand. Particles play a big role in the ocean's biological carbon pump, serving as a reservoir and vector for carbon and energy in the system. We care about particle size because so many relevant particle properties are related to it. For example, sinking velocities and carbon content of particles. NASA and exports is focused heavily on understanding particle size distributions in the oceans because they can be observed from space. So this approach is an important part of our goal to quantitatively link remote sensing observations to the export and fate of ocean primary production. In the exports project, we had the opportunity to make measurements of the concentrations of particles with a large number and variety of different instruments. These instruments generally fall into one of 2 different groups, those that either detect individual particles, such as those on the right, or those that evaluate the ensemble of particles in the water and use those to derive a particle size distribution, such as those on the left. Some of these measurements were done on board the ship with discrete water samples from the CTD rosette, while others were measured in situ by attaching the instruments to the rosette and doing vertical profiles. We are able to retrieve and visualize particle size distributions across an extremely wide range of sizes, spanning up to 6 orders of magnitude. This plot shows an example of one such size distribution intercomparison, and we found that the results agree with each other with a median difference of less than 30% between instruments. The UVP is one rosette-mounted instrument that allows us to measure these particle properties, and it is lowered through the water column and takes images of particles and plankton and stores the images that are larger of particles that are larger than 500 microns in equivalent spherical diameter, and can measure particle sizes and abundance down to about 100 microns. In this example, we see the particle distributions that we can obtain from the UVP, and we see a particle concentration maximum in the upper water column at about 45 meters below the surface, and below this it quickly attenuates to intermediate particle minimum at about 130 meters below the surface, before increasing again to a particle maximum at 250 meters. This intermediate particle maximum is correspondent with a minimum in the particle size distribution slope, which means that there's a predominance of small particles where this maximum in particle concentration is. And so this contrasts with the surface waters where we saw a strong abundance of large particles, very large particles. Given the UVP's ability to capture images of particles and plankton through the water, we are also able to use machine learning techniques to categorize and then manually validate these images into their different taxonomic and particle type categories. And here's just a few examples showing the vertical distributions of crustacea and mollusks, and showing their Diel vertical migration patterns and how they relate to, for example, the abundance of cylindrical fecal pellets that we think are coming from these crustaceans. Moreover, we are able to track these Diel changes in the overall particle concentrations, and we can see slight shifts in the total particle abundance, the total particle volume, and the slope of the size distribution as these plankton communities migrate up and down and influence the sizes and concentrations and repackage particles as they feed and defecate. So in conclusion, these optical methods give us important insights into the dynamics and distributions of particles within the water column and help us make the connection between overlying satellite remotely sensed data products and the export flux that's coming out of the system.
Hi, my name is Hilary Close. I'm an assistant professor at the University of Miami, and today I'm representing a group who's been thinking about the synthesis of data related to the characterization of sinking and processing of particulate matter over depth in the water column. We're using complementary methods for the assessment of physical processes and functional composition of particles obtained by in situ pumps, optics, sediment traps, and the marine snow catcher. I'll talk a little bit more about the different data types and the people involved on the next slides. Starting with this cartoon, we've sketched out some rough size classes of particles characterized by different methods, as well as a coarse division of important zones vertically in the water column. Now, it's worth noting that the size classes here can be different from the size classes characterized by a method like the UVP. Some main points of synthesis we can summarize here. First, between the upper and lower euphotic zone, we see a transition between POC dominated by small phytoplankton particles to a large component of fecal pellets. In other words, by the time particles settle out of the euphotic zone, they've been reworked and or repackaged through partisan and metazoan food webs. Coming out of the euphotic zone, all of our methods show shifts in key processes in the layer just beneath the euphotic zone, so from around 100 to 200 meters. This includes an area of intense disaggregation, which along with direct remineralization plays a large role in the attenuation of carbon flux in this depth range. So delivery of POC into and throughout the mesopelagic is heavily modulated by disaggregation and also by microbial reworking of those disaggregated particles. Carbon fluxes here are low, so mesopelagic food webs appear to rely heavily on small, microbially reworked particles for nutrition, possibly also providing a pathway for repackaging of this material into newly formed fecal pellets that then join the sinking flux. Next, I'll briefly show some example data figures from these groups. First, Vinicius Amaral, a PhD student at UC Santa Cruz, Olivier Marchal and Phoebe Lamb are using particulate organic carbon concentration data to estimate particle cycling rates in the upper water column. The data used are size fractionated POC from pumps and total POC from beam transmissometry, or C sub P, which you can see here in the depth profiles. Particle cycling rates are then estimated by POC data inversion, both in the euphotic zone and a layer in the upper to mid mesopelagic zone. In the model, they include rates of particle production, settling, remineralization, aggregation, and disaggregation. And the schematic here shows that POC budget for the mesopelagic zone. Next, we're synthesizing observations from multiple exports projects to estimate particle sinking speeds as a function of depth. There are a lot of different groups involved in this synthesis, and Meg Estapa, now at University of Maine, is the contact person. This figure shows preliminary results of mean sinking speeds for particle populations defined by direct measurements with the marine snow catcher, inversion of size fractionated POC concentrations, and comparisons of trap fluxes to standing stocks of particles at the same depth. Considering the whole particle population, including the non-sinking fraction, the estimated mean speeds are less than 10 meters per day on average, and the different methods, which you can see here, produce relatively consistent results. Direct measurements of specifically sinking or large particles are here in gray and orange. These are more variable, but generally higher, as one might expect, with mean speeds in the low tens of meters per day. We're continuing to estimate sinking speeds from observations with higher particle size resolution. Next up is the work of Colleen Durkin and the sediment trap team. They have used the imaging of sediment trap gel layers to identify 9 categories of particle types and to model their carbon fluxes and attenuation using algorithms based on size and abundance. The combined modeled fluxes of the particle types shown here in depth profile reasonably compare with chemical measurements of bulk POC flux into the traps. Particle attenuation with depth appeared to be mostly controlled by the type of particle rather than the size of particle. For example, large, long, cylindrical fecal pellets attenuated quickly in the upper mesopelagic, possibly disaggregating and fueling mesopelagic food webs. On the other hand, salt pellets showed no evidence of attenuating with depth. There's also evidence of particle repackaging in the mesopelagic, such as the increase in short ovoid pellets, typical of organisms such as larvaceans, which live in the mesopelagic and filter feed on small particles. Finally, this is the work of my group at University of Miami and Brian Pope at University of Hawaii. We're characterizing particles captured by pumps and traps, as well as zooplankton biomass using natural abundance isotope techniques. This figure shows source amino acid delta N15 values in zooplankton in the black symbols as a function of depth, and the same type of data for small 1 to 5 micron particles highlighted in this red field, and larger and sinking particles highlighted in this blue field. These source amino acid values in zooplankton identify the origins of particles at the base of their food web. In the surface, it appears all particle sizes are important, but coming into the mesopelagic, 1 to 5 micron detrital microbially reworked particles increasingly form the base of the zooplankton food web. We speculate that small particles enter the mesopelagic food web via protistan grazing. And we're using additional information from compound-specific isotope analysis to model the phytodetritus, fecal pellet, and microbially reworked composition of particle size classes and sinking particles. All of these groups are happy to discuss our work further, and we'd also like to acknowledge that our funding on these projects has come both from NASA and the National Science Foundation. Thanks.
Hello, I am Muntsa Roca Martí, a postdoctoral investigator at the Woods Hole Oceanographic Institution. In this talk, I will present a brief summary of how radionuclides were used during exports to quantify sinking particle flux. I will focus on the disequilibrium between thorium-234 and uranium-238. Thorium-234 is constantly produced by the decay of uranium-238 in the ocean, and importantly, the biogeochemical behavior of these two radionuclides is very different. While thorium is particle reactive, uranium is conservative in seawater. That's why thorium gets attached to particles, and if particles sink, thorium sinks with them. This creates a deficit of thorium with respect to its parent in the upper ocean, which we integrate to calculate thorium fluxes. A similar approach can be used for polonium-210 and lead-210, but the time scale is different. Thorium integrates over 1 week to 1 month, while polonium integrates over a few months prior to the sampling period. During exports, the thorium team collected samples at an unprecedented high resolution, almost 1,000 samples over less than a month. Here are the results from 61 seawater profiles. On the left, the color lines show thorium activities for each epoch. The black straight line represents uranium, and chlorophyll is on the right. The area of net thorium removal overlapped with the area where primary production was occurring. Thorium reached equilibrium with uranium between the depth of 1% to 0.1% photosynthetic active radiation. This is a specific station where again we see the thorium deficit area, but we also see excess of thorium due to particle renormalization or disaggregation. Very similar patterns with that were observed for polonium and lead. We also collected size fractionated particles by large volume filtration using in situ pumps. This allowed us to convert thorium and polonium fluxes to POC flux by using appropriate ratios of POC to thorium or POC to polonium on particles. Here is an example of profiles of carbon to thorium ratios, which show a sharp decrease with depth due to the preferential utilization of carbon by heterotrophs. With that, we can derive POC flux, and here is the result. The gray shading shows the cruise average POC flux using thorium, and the dots show the POC flux using polonium at 3 specific stations. Maximum POC flux was measured at 50 meters, which rapidly attenuated down to the depth of 1% par. Interestingly, the good agreement between both radionuclides suggests that fluxes at Ocean Station Papa were close to steady state over the past few months. In addition to POC, we also estimated fluxes of other important bioelements. Our dataset shows a strong attenuation of soft tissue-associated fluxes. POC, nitrogen, and phosphorus are shown here in gray, blue, and red, while particulate inorganic carbon and biogenic silica fluxes were within uncertainties from 50 meters to 500 meters, here shown in black and green. Now we can assess the transfer efficiency by dividing the fluxes at 220 meters by those at the base of the photic zone. As an example, here I show POC, biogenic silica, at all the stations sampled for thorium during exports. We observed lighter colors for POC, which means lower transfer efficiencies or higher flux attenuation for POC compared to biogenic silica. So let's put Ocean Station Papa into context. This plot shows the export efficiency, the fraction of net primary production that is exported from the photic zone, as a function of the transfer efficiency. The size of each circle is indicative of the magnitude of net primary production, and the percentages are the product between the export efficiency and the transfer efficiency. The orange circles here show the results from exports obtained by using thorium and traps, which are very similar. Using thorium, we obtain an export efficiency of 13% and a transfer efficiency of 61%. This places Ocean Station Papa as one of the more modest POC flux settings in the world ocean, similar to the oligotrophic sites Aloha and Bats. In the future, we will analyze station-to-station variability in relation to other exports datasets to better understand the underlying controls on the biological carbon pump. So there is more to come. Thanks for your attention.
Hi, I'm Rue Nicholson at the Woods Hole Oceanographic Institution.
My name is Shani Traylor, and I'm a PhD student in the MIT Woods Hole Oceanographic Institution Joint Program.
And I'm Alex Niebergall, a PhD student at Duke University. Along with Brandon Stevens and Muntsa Roca Martí, we have been leading synthesis efforts of biogeochemical budgets on exports. I will start by defining some of the biogeochemical rates that we will be discussing today. As we know, in the surface ocean, photosynthetic organisms use a combination of carbon dioxide and other nutrients to create organic matter and oxygen. The total organic matter produced in this reaction represents gross primary production, or GPP. Some of this energy harvested through photosynthesis will be used for autotrophic respiration, and the net resulting measurement is called net primary production, or NPP. When we then factor in heterotrophic respiration, the resulting measurement is net community production, or NCP. We can measure these rates by measuring the production and consumption of different constituents in this equation. Here I present preliminary results comparing 10 data sets of NCP and new production in the mixed layer during the course of the exports field campaign. We collected O2 argon measurements using equilibrator inlet mass spectrometry on both ships. We have oxygen budgets from 3 sea gliders. We have 3 data sets from the BioArgo float using oxygen, nitrate, and DIC and total alkalinity. We have N15 incubations on the Roger Rebelle and beam attenuation measurements from the Wire Walker. As you can see in this map, these data cover approximately 100 by 100 kilometers, with some measurements being Lagrangian and others Eulerian. In this figure, the measurements are color-coded by their platform and organized on the x-axis by tracer. The gray shading represents historic measurements of summertime NCP at Ocean Station Papa by oxygen-based BioArgo measurements. As you can see here, though the measurements have different ranges, the median of each data set aligns well with the historical measurements, with NCP generally being relatively low but positive throughout the course of the cruise.
Over annual timescales, net community production is balanced by the carbon export from a given depth boundary. To complement our cruise measurements, we leveraged numerous autonomous assets to estimate the long-term NCP integrated to 100 meters depth. 100 meters is a typical depth choice that allows for comparison to instruments that measure particle fluxes, such as sediment traps. The assets included gliders, biogeochemical Argo floats, and a mooring, which sampled the northeastern Pacific as seen in the map below. The climatology from 2008 to 2020 was also constructed from a combination of float and mooring data. On the right is the time series of NCP integrated to 100 meters in units of millimoles of carbon per meter squared per day. The oxygen-based results show good agreement in both the timing and the amplitude of NCP, with a switch from net autotrophy to heterotrophy from the late fall of 2018 to the early winter of 2019. The nitrate-based results show similar timing, but with larger amplitudes, and the DIC-based estimates closely follow this as well. These longer-term estimates show a similar dip into heterotrophy in the fall of 2019. Overall, we see fairly good agreement between all these different approaches, given the variability and uncertainty associated with each estimate.
Below the euphotic zone, in the mesopelagic zone, exports measurements allow us to quantify carbon budgets for the zone from 100 to 500 meters. A range of measurements, including from traps, tracers, and numerical models, result in a supply of organic carbon to the mesopelagic of 1.8 to 3.5 millimoles carbon per meter squared per day. The supply from above supports respiration and the consumption of carbon from below. Estimates of this carbon demand term were from 4.8 to 9.7 millimoles of carbon per meter squared per day, and that's more than double the supply during the study period. And these tracers from biogeochemical measurements, incubations, and model results indicate a system that was not in steady state during the time of the exports experiment. And this means that the consumption in the mesopelagic relies on either carbon that was exported earlier in the season or from advective inputs of organic carbon.
Okay, I'm sure you can all see we've had a little bit of a chat going on in here about the Irina asked a question about the smaller size particles at intermediate depths. And we're talking about zooplankton repackaging as well as particle fractionation. I don't see any other questions at the moment. Oh, here comes one. Why is there first a decrease and then an increase of biogenic silica flux with depths? Assuming that figure was perceived correctly.
Yeah, so I can answer this. So actually in that figure, we can see a little bit of variation in the biogenic silica flux with depth, but if you consider uncertainties, these changes are not that significant. So yeah, that's what it seems that is decreasing. But if you take a closer look at the uncertainties, the fluxes are not different within uncertainties across the different depths.
Again, just a reminder, make sure your questions, if you're putting them in the chat, go to everyone. Here's a couple right here. Cindy, no direct measurements of particle density. Okay. Did Cindy ask a question that I haven't been able to see? Let's ask this one. For Amy Moss and Andrew McDonnell, wait, so you're saying above that particles observed by Andrew are bigger at the surface, then as you go deeper in the water column, they become smaller because they're eaten up and excreted by zooplankton or filter feeders. And then again, particles become larger. Does this point specifically where zooplankton or filter feeders live? I'm going to let Amy or Andrew take that.
Yes, we think so. So there's a couple different lines of evidence that are all coming together that are showing that there's some definite transformations that are occurring all sort of at a similar layer. We see it in the particle size distribution. We see it in the distribution of vertical migrators and also organisms that we think are likely to be doing repackaging, as well as a little bit in the isotopic data on the particles. So there's a couple lines of evidence that are suggesting that, yes, looking at that particle size distribution is helping us see where those transformations are occurring. Again, there are some other physical processes that can play a role there, but these lines of evidence do seem to indicate that that's what's happening there.
Is there any evidence of lateral gradients in POC to DOC that could close the carbon budget given the low advective velocities in the area?
Hi, Heather, this is Rue. Maybe I can take that one. So we have been looking into this a bit, and we think it's not the most likely cause for closing the carbon budget. There is some slight correlation between isopycnal spice and DOC in the mesopelagic that we're investigating, but I think it's more likely that this is a temporal mismatch, and we're thinking more about suspended small particulate or dissolved organic carbon that may have been exported earlier in the spring or earlier in the season that's getting consumed.
Thank you, Rue. For Hilary, it has often been stated that sinking matter is extensively transformed in mesopelagic ocean. How did the integrity of sinking particles look like with what did the integrity of sinking particles look like with depth from the sediment trap gel samples?
Yeah, so I don't know if Colleen is on here. If she is on the chat, she might be able to say something about that. But I know that she does one of her categories of particles that she enumerates is basically aggregates, something that can't be exactly classified as an identifiable fecal pellet. So that could be the category to look at to ask that question. And then I guess I'll just say, you know, this is a reason why we're also, you know, trying out chemical methods to look at the composition of the particles, because when they do get morphologically changed, can we still identify their source from some aspect of the chemistry? And so far, you know, we're working on putting these different data types together, but so far we do think we have a pretty robust indicator of fecal pellet content through the isotopes. So I think, you know, we're still working on putting that right side by side with those morphological characterizations that Colleen has been doing. And I see that she is responding.
I've just unmuted you, Colleen, if you'd like to respond with your voice.
Oh, well, yeah, so the deeper traps, most of the particles attenuated with depth, but the deeper trap samples didn't have very many fecal pellets that were intact. It was mostly these kind of translucent aggregates and other dense detritus, and I really think most of that came from degraded fecal pellets.
Other questions? Have people studied the PIC fluxes also?
Actually, yes, we were measuring PIC concentrations on our particle size, our particles in different particle size fractions. And yes, we have calculated very low PIC fluxes. So if we look at our PIC concentrations and our ratios PIC to POC, and we compare those to previous studies, our PIC data is very low. And actually, if we have a look at satellite data, we can see that when we were there in 2018, August, September, the average PIC concentrations in surface waters are a factor of 2 lower than the typical values in August, September during previous years. So our PIC fluxes are very low.
Other questions? Let's see, I think I've got one in the Q&A here. Just to loop back, because I'm not sure if everybody saw it, Cindy Lee had asked a question about whether or not particle density was being measured, and the answer is no, there are indirect measurements, and you can see that in the chat here. So Cindy asks, great information on particle size and sinking rates, but is anyone estimating or measuring particle density? And I will read the response. Megastopper responded, Cindy, no direct measurements of particle density. We might try to constrain this indirectly, depending on precision of sinking speed estimates from well-constrained particle observations, which is still in progress. Just wanted to make sure everyone could see that. Other questions? Looks like we have another one. Here we go. Do we know the mechanisms by which the disaggregation in the deeper part of the euphotic zone occur?
So this is Amy. We did not do very many specific experiments on board to try to characterize that. So we're in the same role as everyone else, which is trying to trace different lines of evidence in to point at it. So we have no real smoking gun, and we think that that's probably a suite of things, but we don't have the mechanisms nailed down.
Okay, going back to the density, the particle density question, lots of number concentration data were shown, but do we have a good idea at the surface at Papa what dominates biomass, autotrophs, heterotrophs, and what size class?
I can address part of that. So from our size fractionated POC and chlorophyll data, we know that at the surface, the majority of the community is in the less than 5 micron size class. Someone else may be able to address the autotrophic versus heterotrophic a little more than I can.
Okay, and
Yeah, hi, this is Jason. I would say that we're sort of just at the point where, I showed the phytoplankton size distribution and the biovolumes we can get for different size classes. And we're just, I think, at a point where we're starting to put all of the different contributions from the groups together. And I think eventually we will have a good estimate of which groups and which particle types contribute to the total POC or biovolume or total biovolume.
thanks, Jason. From Colleen Petrick, can CSIA elucidate feeding relationships like feeding on plankton directly versus detritus derived from those types of plankton?
Yeah, so that's me. Thanks for the question. So I guess it depends on how deep you're trying to go, because a living versus a dead phytoplankton is not going to look different isotopically, but if we're talking detritus that has been microbially reworked, we do see some changes in various aspects of the amino acid isotope ratios. So right now we're actually working on characterizing end members. So we're characterizing phytoplankton end members, fecal pellet end members, and then we're even trying to see how microbial reworking alters that fecal pellet signature. So yeah, it's those are pretty much the end members that we can look at right now. But if you're talking about living versus dead phytoplankton, we can't get that specific. But we are also, one other thing to mention is we're also working on elucidating the protistin stuff in the food web. So I was showing some data on zooplankton as well as particles, and we do have ways that we think that we can get at how many steps are in between those. So basically estimating the length of the food web. So that might also get at your question a little.
For Andrew, do you have an estimate for the proportion of dead versus living particles in your imagery? And do you expect that the size spectrum slope will be able to translate from the size ranges measured from the UVP to smaller size ranges? For example, the less than 10 micron range.
Yeah, that's a good question. We just recently finished sorting all of the large images from the UVP. So this consists of particles larger than 500 microns in ESD. And I don't think I have the data in front of me, but a lot of them were non-living, we think. And so the living were maybe about half of them or a little bit less than that. But it depends greatly on what depth we were looking at. A lot of the main targets were in the upper water column, were living probably, and long fiber-like particles. But what was the second part of that question? And I guess maybe by combining some of the methodologies, especially with maybe the IFCB, for example, we can begin to fill in that living versus non-living fraction across a wider range of the size distribution. But the UVP on its own can only really distinguish those two above 500 microns.
I feel like every time I
follows.
type, I mess something. Yeah, I mess something up every time I type.
Go ahead
I know,
and go ahead and say it.
Yes, it's so much easier. So the same way that you can sort of, we want to see whether the particle size distribution goes smaller and is predictive smaller. We also want to see if the particle size distribution goes larger as well. So there's an overlap in what the UVP and the mock nets sample. But as you start to get to larger size particles, they become more rare and you're less likely to sample them with the UVP. And so in that way, we're taking our net samples and we're putting them on a ZooScan, which gives us a very comparable kind of data set to the UVP, but is more quantitative in the larger size community. And so we are doing the same thing where we are sorting live and dead in ZooScans and then pairing that up particle size distribution with the UVP to also see if we can go forwards to the large size class as well as trying to go backwards to the smaller.
Any more questions today for our speakers? Okay, I think we can progress to Dave's closing remarks and updates on the field campaign.
Okay, so what's next for the exports field program? So here is shown kind of the overall program timeline for the exports field campaign. There's basically two important phases. There's a field work component that we're in the middle of right now, where we have the North Pacific cruise that we heard about that we did in 2018. And we had planned to do a cruise to the North Atlantic in the spring of 2020. That, if those are both successful and data are put up, the plan is for NASA to compete a phase 2 program on data synthesis and modeling to answer basically the third question of exports. How can we use this knowledge to improve our predictions of present and future states? The problem, obviously, is we didn't get to do our cruise this year. We got COVIDed. And what NASA has done is it has extended the period of phase 1. They've raised funds to do that. And we have good news. We are in the process of planning a cruise to the North Atlantic for basically May of 2021. So what do we want to do? We want to find a contrasting end member to the North Pacific cruise that you just heard about. We want to see higher biomass and export, low flux attenuation, etc. Our focus, we hope, again, the post-bloom evolution, hence we want to do this in May at the PAP site. And fluxes due to zooplankton, the Diel processes should lag that. We hope to see large phytoplankton and lots of diatoms because we need to make Bethany and Mark happy. And this is a region of higher mesoscale and sub-mesoscale energy, which provides both issues in sampling Lagrangian experiment, but also opportunities for understanding more of the role of physical transport, especially on some mesoscales. The sub-mesoscale, the high mesoscale and sub-mesoscale energy will also lead to a much more patchiness in fluxes and biomass than we saw in the Pacific. So our cruise is scheduled for 2021. We were officially postponed by NASA. We are in the process of planning a 2021 cruise in May to the Porcupine Abyssal Plain Observatory of 28 days of sampling above the sailing on the Discovery and the James Cook out of Southampton in the UK. If we're successful here, this will complete the exports phase 1 program and enable a phase 2 to be considered by NASA to release proposals for competition. To make this all succeed, we needed ships, agency engagement, PI commitment, and a solution to the pandemic. Thanks to the efforts of many, we really have solutions to the first 3. I'm very excited and a little startled, to be perfectly honest. We're hoping that we can get a workaround or a solution to the 4th one of these issues early next year. So with that, I'd be happy to take questions, and thank you very much.
Okay, well, I for one am thrilled that we are able to reschedule the field campaign in the North Atlantic. We have a few minutes left if anyone has Any remaining questions about what Dave just talked about or anything we didn't get to in the last vignettes? When can we expect phase 2 RFP?
Okay. I suppose this is mine to answer. I'm not a program officer.
Is it yours or mine?
So the plan is to, once the North Atlantic data is collected, that there will be an opportunity to set up, what this phase 2 would be, is it sort of a data synthesis and modeling portion of this, aiming at this third question about how do we use the data that was collected here and other data sets to understand what to reduce uncertainties on how the biological pump functions. Everything's kind of if you haven't noticed, everything's kind of messed up right now, and we will see how things go forward. We do need to complete the North Atlantic cruise to allow that. The grants were extended, at least all the NASA grants were extended until like February of 2022. So it's not likely that that a call for proposals won't be any earlier than that summer. We would have to present, all the data has to be publicly available so everybody has a fair shot at competing for this, if that makes sense. So that's what it is. Everything's, a little bit of uncertainty kind of stinks. And we really, we've got to get past, all the COVID stuff. So we will see. But that's the plan at the moment.
Hi, can I jump in? Hi, this is Yvonne. I'm going to offer a shorter answer than Dave. Just for insurance data policy, everybody has to submit their data, and they have 1 year after collection of data to submit the data. So if you're doing a field in May, that means May 2022, we should expect majority of the data to be inside the data set, and then not prior to that. So if you want to plan, just don't plan anything prior to December 2022. If you have any other questions, you can direct them to Laura Lorenzoni, who's the project manager of this gigantic thing.
I'm gonna pose another question, and then I will actually go in and see if Laura's still on and unmute her in case she wants to follow up on that. There was a question. She's not on right now. Okay.
I think she is. Yeah. Sorry, she has to leave. I killed my child so I can answer this question. So she's probably
So question about remaining COVID issues. If there's not a vaccination, will the crews be able to move forward? I'm sure there's uncertainty about that, but if you have any insights.
Do you want me to take this, Dave?
So let me just say, we will not do anything that puts people into harm's way. So we are working, every institution to do travel has their own requirements that have to be met. We have to meet NASA's requirements and the requirements of the Natural Ocean Center. So we're working with them to come up with some sort of process that allows us to reduce risk as much as possible. Everything's changing. We had a couple hours last week talking to them, and we're probably going to have a couple hours this week. We will figure this out. But the important thing to know is that we will not do anything that will put people into situations that they're uncomfortable with. We don't think that is correct, and we have to do this as safe and as risk-free to the individuals participating as well as the agencies and the agencies investing. So.
Thanks, Dave. It looks like one question, a final question here. Is there any intention to link this with or use this to shape the nascent Biogeoscapes program? I'm not sure if anyone on the line can answer that, but I can tell you there's a lot of PI crossover between exports and those who are starting to help shape that program. So I anticipate a lot of links and crossovers. But if anyone wants to follow up on that. If you message me in the chat, I can unmute you if you happen to be on the attendees. I anticipate there being a lot of crossover just because there's so much PI crossover. And it is a huge multidisciplinary field campaign. So obviously it provides a blueprint. Any other final questions before we wrap up today? I want to thank everyone again, especially the speakers who put together these fabulous presentations, and the organizers, Ivona and Dave and Amy Moss and May, who put these all together and made them work. I just want to say a thank you to everyone. It was so great today to be able to see so many different facets of the exports campaign and the data that are coming out of it presented in one place. If anyone has follow-up questions, the speakers would be happy to hear them after the webinar is over, or you can send them to me and I can make sure that the speakers receive them. I want to thank everyone again. We are hoping to have one more talk in the OCB webinar series in this calendar year, and just stay posted and we'll let you know when and what topic that will be on. Thank you everybody again, and stay safe and well, and we look forward to seeing you again soon.